Okay, good afternoon, everybody, and welcome back to our microbiology class. We're continuing now with this current set, PowerPoint set, which will probably last this class and maybe the next class as well. And we're mainly now discussing cell structure, concentrating on prokaryotes, of course, but we will address eukaryote cell structure a little bit as well towards the end. But don't forget that a lot of the principles that apply to the prokaryote cell also apply to eukaryote cell. And our first topic is a case in point. Because we said at the very beginning, all life is based on the cell. This is the basis of the cell theory. All life is based on the cell. All cells are produced from another cell. And right at the beginning, we said the defining feature of the cell, no matter what kind it is, no matter whether it's a prokaryote cell or a eukaryote cell, the defining feature of the cell is the membrane that surrounds the cytoplasm. You'll see it given various names: the cytoplasmic membrane. Plasma membrane is now the commonest designation, but you'll also hear it referred to as the cell membrane. And for some reason or another, that's fixed in my brain. Just remember that those three terms are equivalent: cytoplasmic, plasma, or cell membrane. They all mean the same thing. So it's an extraordinary thing. This membrane around the outside of the cell is in fact a liquid. It is made up of an oil. It's made up of a lipid. And it is in fact only 2 molecules thick, as we'll see in a second. And yet, despite that, it forms a very profound boundary. And this is a boundary between the outside of the cell and the inside of the cell. And because it is such a powerful boundary, it allows the inside of the cell to be different, for the conditions to be maintained differently to the conditions outside. It allows the cell to control the constitution of the cytoplasm, and we'll see exactly how that's achieved in a minute. So we look now at the basic structure of this membrane. And remember that in the prokaryotes, this is the only membrane. We only have the membrane around the cytoplasm. But the same principles apply in the eukaryote when we consider the membranes that surround the organelles inside. They have much the same structure, the same principles apply, as we'll hear. So the very first thing to understand is that the fundamental unit for making up this membrane is this. Just 1 second. I just want to do something. There. The fundamental unit making up this membrane is this molecule here. Now it looks very complicated, but in actual fact, it's quite a simple molecule. And this is a phospholipid. This portion here, these long chains, those are hydrocarbon chains. They are lipids, they are oils. And this part here, this is actually alcohol sugar, but this whole thing here is a phosphate. Just never mind all the complications. Just think of this as being a complex phosphate-rich group that is sitting here, and it's binding these 2 long chains of lipid here. So this molecule has a very strange composition, because these lipid tails here are strongly, strongly hydrophobic. They repel water and are repelled by water very powerfully. But these heads here, these phosphate heads here, the phosphate-rich head is very, very hydrophilic. It is where these are hydrophobic, they're repelled by water. These are hydrophilic. They dissolve in water very easily. They remain attached to these lipid parts though. So when we take a whole lot of this phospholipid and we put it in contact with water, if we treat it in certain ways, I'll describe it in a little bit more detail in a minute, but the only way that this can organize itself so that it satisfies all of the properties of hydrophobicity and hydrophilicity is to organize itself like this. If this is surrounded by water, there's water out here and there's water here. Here's how these molecules organize themselves with the hydrophilic heads that dissolved in the water on either side, and with the hydrophobic tails all pointing inwards like this. And by doing this, they exclude all the water from here. So there's no water that satisfies the hydrophobicity, but these are happily dissolved in water on the outside. And this is the basic constitution of this plasma membrane structure. It forms two, you can think of them as being two leaflets, a sandwich put together like this, with the interior completely hydrophobic and free of water, and then the outside heads here dissolved in the water. It is in fact a liquid. These molecules can actually move around quite freely, but they associate very strongly with one another through various weak molecular forces. So this, even though it's liquid, it forms quite a robust membrane structure. And just as in its simplest form, as we're looking at it like this membrane will only allow certain things to pass across it. Water molecules can just move across, believe it or not. They can get across very, very slowly. Some ions can also move across, but they don't do so very easily or very rapidly. Most other molecules actually cannot move across. So they just cannot move across. So immediately, this is a barrier. This is a barrier around the cell, protecting the integrity of the cytoplasm on the inside. Here, you can see here, this is an electron micrograph of this membrane. And the space in the middle here, this is all the fatty acids facing into one another like that. On the outside here, these dark lines, these are these phosphate heads, sugar phosphate heads.
Look to my left, look to my right, and like what I saw.
Love you in that scarf. Why do you wear scarves?
Excuse me.
That place?
Hello? Can you mute, please, and not watch other things while you're trying to watch this? Thank you, whoever you are. Okay, so that's where we are so far. Now, if this was all there was around the cell, right, this wouldn't work very well, because there's no way for stuff to, we need to be able to move molecules across. We need to be able to get them to move in both directions. The cell is going to want to send molecules out, and it's going to want to pull molecules in. Just like this, the membrane wouldn't work very well, but that's not how it's actually structured. Instead, the membrane, we're going to see, has portals in it. It has ways of allowing molecules to move through under control. And this is the key feature of this membrane. It affords the cell control over what goes in and over what goes out. Because certain molecules move across quite easily and others do not, we refer to this membrane as being semi-permeable. It does allow things to go across, but there is a selectivity to what moves. And we'll see in a while that actually in the structure of the membrane, water is going to move across quite easily. Not because of this, but because it has gateways for the water to move. Other things like ions, this sort of thing, they do not move across easily. Other molecules do not move across easily at all. And the cell exerts very careful control over what moves and the way in which it moves. So the take-home message about the cell membrane is it allows the inside to be different to the outside. And it allows the cell to concentrate molecules. For our biological reactions that we're going to talk about, some of them very, very complicated, we're going to have to localize reactions. And we're going to have to concentrate substrates, for example. This allows the cell to do that inside the cytoplasm. It also allows the cell to begin to use this membrane as a site for various reactions to take place, as we'll see in a minute. So here's the actual structure. First of all, notice here's our basic membrane structure. These two phospholipid leaflets sitting opposite one another like this. But embedded in this matrix of phospholipids, there are proteins, many, many proteins. They actually float in it. They can actually move around. If need be, the cell can actually actively move them to particular areas if particular activities have to take place in one particular place. So these are the proteins. Some of them span the whole width and go from the outside to the inside. And those we call integral proteins. Others are peripheral. That is, they sit, and they usually, most of the peripheral proteins, but not all, sit embedded on the inside here, partly in the membrane and partly in the cytoplasm. But there are some peripheral proteins which are embedded partly in the outside and partly in the membrane. So when we look at these proteins are very, very diverse. Their structure and their function is incredibly diverse. They do many different things. In the prokaryotes, for example, they are responsible for cell wall synthesis. They are responsible for all of the energy reactions of respiration that we're going to talk about when we talk about metabolism. They all take place on the membrane. In those bacteria which are able to photosynthesize, photosynthesis takes place on the membrane. They are also sensory. I'll explain that by saying to you that this little cell, just a cell on its own, it has no eyes, it has no ears, yet it needs to know what is happening in the environment. And the bacterial cells are capable of sensing many different environmental features. They can sense pH, temperature, presence of nutrients, presence of things that are bad for them, et cetera, and they can get the cell to respond to those conditions in certain ways. Though they are able to do so because there are many sensory proteins which are embedded in the membrane. And those sensory proteins are picking up particular pieces of information. And once they pick that information up, they relay the information to the interior of the cell, and they get the cell to do certain things. But on and on. There are many, many, many different kinds of functions that these proteins will show. But probably the most important of all of the protein functions is this issue of maintaining the conditions within the cytoplasm and keeping them optimal and bringing into the cytoplasm things which are useful to the cell, expelling things which are not useful, waste products, etc., and performing all of these features of transport. In other words, the interior of the cell is continually communicating with the outside, but it does so in a controlled fashion. And that control is exerted by the proteins which are embedded in it. Now, there's a particular way of referring to this model. This is called the fluid mosaic model. Fluid mosaic, because this is actually fluid. The membrane itself is a fluid. And it's a mosaic because it is embedded with all of these different proteins. And the proteins move within that fluid. It's not a static thing. It's a dynamic and living part of the cell. Okay, so we are going to talk more in a little while about the transport functions on the membrane. So the next thing to remind you of, I showed you that outside of the cell membrane, the plasma membrane, that we can have many different structures, but they in general do not participate in these kind of transport functions. These are mostly inert features of the outside of the cell. And the first most important one to think about for the moment is the cell wall. And the cell wall is completely distinct from the cell membrane. It is not a living entity. It is constructed by the cell under genetic instruction. So it adopts a certain form. It's the cell wall which gives the cell its characteristic shape. It's also not a static thing. It is the molecules of the cell wall are continually broken down and rebuilt so that the cell wall, for example, can expand to allow the cell to grow. The cell, it provides a cell shape, it provides structural support, but its most important function of the cell wall is something that might not be obvious. Let's just go back here for a second and think about the cell membrane. I told you that the cell membrane is selectively permeable. One of the sets of proteins that are embedded in the cell membrane is a set of proteins called the aquaporins, gateways to water, the aquaporins. And the aquaporins allow water to pass relatively freely into the cytoplasm. Now you think about it this way. The cytoplasm is a concentrated solution of many different things. And the laws of diffusion tell us that things will move from the areas of high concentration to low concentration. Think about it this way. Where is the water concentration high? The water concentration is high outside. The water concentration is low inside, and water will continually pass from the outside to the inside of the cell through these aquaporins. So there's a grave danger here, because as the water passes into the interior of the cell will begin to swell. Now, this is a problem, especially in fresh water and many different situations. This is actually a problem. Eukaryote cells often solve it by actively pumping the water out, but prokaryotes don't have that kind of facility. And they're under grave danger of actually swelling to such an extent that they burst because of the pressure of the water building up inside. This diffusion of water has a specific name, and it's called osmosis. So the water passing into the interior of the cell causes an osmotic pressure. And the cell has to do something about that. The cell has to find some way in which to deal with the swelling. And the principal way that prokaryotes do it is by building this cell wall. Because what happens is that the cell, as water passes into it, the cell swells up, and it swells up until it hits the cell wall. And then it swells against the cell wall, but the cell wall stops it from swelling anymore. And once that point is reached, then the pressure within is the same as the pressure outside. Think of it as the cell wall is imparting pressure. And now the osmotic pressure is equal on both sides. As much water passes in will pass out, thanks to the cell wall. But it's a completely inert process. The cell doesn't have to, once it's built the cell wall, it doesn't have to exert any more energy in order to regulate the osmosis. So this is the most important function of this cell wall, is that it prevents the bursting of the cell under osmotic pressure. That bursting, by the way, is called lysis. You can do this experimentally quite easily. You can actually dissolve off cell walls, of cells, and you can just have the cells floating around, just the cell membrane. They're perfectly fine. The cell's perfectly functional. But if you put them into dilute solution, like fresh water or something like that, the water pours into the cell, they swell up, and you can see it happening. You see them swell up and then they burst. Without the protection of the cell wall, they burst very easily. So this has some clinical significance for us. The cell wall, in actual fact, does have some pathogenic quality to it, some clinical significance, because our immune system actually reacts quite strongly against some of the components of the cell wall. But clinically, one of the most important features of this is that many antibiotics, such as penicillin, operate by stopping the cells from being able to build the cell wall properly. These antibiotics weaken the cell wall, and as a result, the cells burst. And that is how antibiotics like penicillin actually work. We also have a natural system. In tears, in saliva, in mucus, we possess a compound called lysozyme. It's actually an enzyme, and it's an enzyme that breaks down the cell wall of bacteria. And the same thing happens. The cell walls break down and the cells burst because of osmotic pressure. So this cell wall becomes very important clinically, and it actually, we're going to discover that it leads us into one of the most important of all of the clinical tests that we do, and it reveals a huge evolutionary divide in the prokaryotes, in the bacteria. So let's have a look at this, what makes up the cell wall itself. It is made of a substance called peptidoglycan. That's an important name you must remember. Not so important from my point of view. It's actually made up of the saccharide units, which are joined together. One of them called N-acetyl glucosamine, the other N-acetyl muramic acid, NAG and NAM. They are bound together one after the other, 1, 2, 1, 2, 1, 2, and they form these fibrils. These very, very strong, very powerful fibrils. And this is carbohydrate, basically sugars, and they form a sort of a backbone. But the backbones are bound together. First of all, they're bound like this. There are strong protein bridges which are formed, which bind them together like this. And then they can be bound in layers, one on top of the other. So here, for example, you can see a whole lot of them all bound together here into a thick layer. And they're vertically through this, tying them together and tying them to the cell membrane is another important molecule. This is called teichoic acid there. Now, this structure that you see here is characteristic of this one evolutionary group of prokaryotes, which we call the gram-positive bacteria. You may already have seen this in the lab. First of all, gram refers to the person who first described this situation, but Gram also developed a staining technique to show whether or not a cell was a gram-positive cell. This is the typical gram-positive cell, a thick layer of peptidoglycan, and the peptidoglycan bound together with teichoic acid. And it binds it, here is the cell membrane here, plasma membrane, whatever you want to call it. Here it is here. This, it's important to note, this thick layer of peptidoglycan is not a barrier at all. Okay, it's got huge gaps in it that allow molecules to pass freely back and forth. There's no control over what passes back and forth. It's not a living entity like the cell membrane at all. It's inert. So there's a couple of clinical things to note. First of all, teichoic acid is also recognized by the immune system, and it can stimulate an inflammatory condition. Second of all, it's this synthesis that penicillin affects. Okay, it's this synthesis of the peptidoglycan that many of our antibiotics operate on. So here it is here, really important to make note, because you are certainly going to be asked about the structures of these different cell walls. Okay, it's important to get them straight in your mind. Notice something. The significance will become clear in a second. There is only one membrane here. Okay, there it is. There's the plasma membrane, and then this peptidoglycan cell wall sits. The thick peptidoglycan wall sits outside. That's gram-positive. Gram-negative bacteria are completely different. The structures outside of the cell membrane are completely different. They still have peptidoglycan. They still have a cell wall, but it performs the same function. It's osmotic control, but it is very thin. There it is there. It's a very thin layer. It has no teichoic acid because it doesn't have multiple layers, one on top of the other. Here's the cell membrane there. Here's the cell wall there. And then there's an outer membrane. That's its name, is called the outer membrane there. And you'll see it looks very much like the cell membrane. And in some ways, it functions like the cell membrane as well. But it has distinct differences. The first is that it has phospholipid in it, but it is also, especially this outer leaflet here, is very, very rich in another type of lipid molecule, which is called a lipopolysaccharide. And the lipopolysaccharides actually have saccharides attached to the lipid group, where before we just had the phosphates. Now you actually have polysaccharides. You have sugar units attached to the outside. This is especially true in this outer leaflet here. But otherwise, it's still this bilayer here. And it also is penetrated by different proteins. And especially importantly, penetrating here are sets of proteins called the porins. And the porins are complex proteins which selectively allow specific molecules to move across this outer membrane here. They move across and they get accumulated here in this space. This space in here is called the periplasm. And in some ways, it's somewhat similar to the cytoplasm in that it's almost gel-like. It's so rich in molecules. And the periplasm is an important store of nutrients, especially. And these bacteria that have this structure are especially good at accumulating rare nutrients. So this is typical gram-negative, the gram-negative cell wall. And I'll explain the naming in a second. So the structure is completely different. The gram-negatives have these two membranes. They have the cell membrane, and they have an outer membrane. And between the outer and the cell membrane is the periplasm with a thin cell wall in it. Now, the gram stain you will do in lab works because when we go to this gram positive, what we're going to do is we're going to actually use a staining technique where we're going to stain the whole cell. We're going to stain it with a stain called crystal violet. And crystal violet fills the whole cell and stains it a deep blue. It doesn't matter whether it's gram positive or whether it's gram negative. It does the same thing, stains them both deep blue color. But then we're going to treat this stained cell with alcohol. And what will happen, there are a couple of other steps which I won't go over, but what happens in the gram-positive bacteria is this thick peptidoglycan binds a lot of the stain, and it prevents stain from washing out of the cell when we treat it with alcohol. And as a result, after we treat it with alcohol, the cell stays blue-black, a blue-black color. In the case of the gram-negative cell, however, we're going to stain it with crystal violet. The whole cell stains, but now we're going to wash it with alcohol. And as soon as we wash it with alcohol, we disrupt these membranes. We disrupt the outer membrane. This peptidoglycan is very, very thin, and it does not stop the stain from washing out. You have to do it quite carefully, but in the gram-negative, all of the stain just washes right out as soon as you treat it with alcohol, and the cell goes colorless. In fact, to visualize the cell, you have to stain it with another dye to see them at all. The blue-black color is lost. So the gram-positive, we say it's positive because it keeps the stain. It keeps the blue-black color. Gram-negatives, they're gram-negative because they lose the stain color, all developed by gram. That's why it gets the name. This is the classic clinical test in the lab. This is one of the very first things you do when you're trying to identify a bacterium. Which of these huge evolutionary groups does an unknown bacterium belong to? Is it a gram-positive or is it a gram-negative? They are very, very different to one another. As you can see, the structure and the function of the two groups is very different to one another. For our purposes, there are important pathogens in both, but I will just mention that gram-negative bacteria are also very common, especially in systems like aquatic systems where nutrients are quite rare, because they're very good at picking up rare nutrients. But they also, where there are many important gram-negative pathogens as well. And here is one of the big problems with the gram-negatives. And that is that outer membrane can cause very, very profound immune responses. Our immune systems respond very, very strongly, especially when these components are seen in the bloodstream. So here, this is one of our lipopolysaccharides. And the lipid part here, which is embedded in the membrane, this lipid part here is called lipid A, and lipid A has attached to it these polysaccharides. And this lipid A in particular is toxic. when it goes into circulation, it is toxic, and it is one of the things which causes this very profound collapse of blood pressure because of an immune response, which we refer to as septic shock. And this particularly becomes a problem as gram-negative cells in the circulation die and disintegrate, and they release these as free molecules in circulation. They tend to cause this very profound immune response. But the polysaccharides as well are also antigenic and are recognized by the immune system. So the gram-negatives, especially when they go into circulation, are a particular clinical problem. Now, I just want to, most of the bacteria that you are going to deal with, especially those that you see in lab, are going to either be gram-positive or gram-negative. There's some groups, though, that are a little bit different, a little bit difficult. And the most important of all of these groups is this genus of bacteria called Mycobacterium. The genus Mycobacterium, there are several different species, but some of them are very important in human health, and not only in human health, but in animal health, let's say. And you'll recognize them immediately. I tell you the names, because Mycobacterium tuberculosis is the causative agent of tuberculosis. Mycobacterium leprae is the causative agent of leprosy, or now more correctly referred to as Hansen's disease. Both of these, very slow-growing bacteria, but extremely toxic, and they cause grave illness, both in human beings and where they occur in animals. There are other species which occur in animals. They can cause severe illness and death. And there's a big problem with the mycobacteria, and that is they are fundamentally gram-positive. They are allied to the gram-positives, but they contain in their cell membranes, they contain a special lipid molecule called mycolic acid. And mycolic acid is actually a wax. And because of that, they are very difficult to stain. You can't take them through the gram stain because the stain just doesn't get into them because of this waxy coating around them. And so we have to use a special staining technique. I think you won't do this, I don't think, but you may see it in lab. We use a special technique called the acid-fast stain procedure. And the acid-fast stain procedure produces a very characteristic red coloring. Here you see them here. This is actually Mycobacterium tuberculosis. And this is in probably a sputum sample from somebody who is suffering from tuberculosis. This is a standard clinical procedure to test a sputum sample from somebody suspected of having Mycobacterium tuberculosis. You do an acid-fast stain procedure, and if you end up with these red-colored stains, it is positive. What you need to remember is that name, mycolic acid. It's special to the mycobacteria. Okay, so all of that section on the cell wall, it's very important. Please make sure that you understand the differences in the actual structure. You are going to be asked in tests about the difference, right, between the two. And do not immediately go, the gram-positive is gram-positive because it gives a positive gram test. You need to explain why it gives that gram test, because of the structure of the cell wall. Okay, so now let's start talking a little bit about the membrane function again. But this time we're going to talk about it in a little bit more detail. and we're going to have a look at how it is that especially the proteins interact with the cell membrane to provide this controllable barrier between the outside and the inside. So first of all, I told you at the beginning, the membrane on its own will allow some molecules, especially small molecules, to drift across. They don't always do so very easily. They don't always do so very fast. But for example, water can get across the membrane. Some ions drift back and forth as well, but they don't do so particularly fast. Instead, if the cell actually wants to move molecules efficiently, it makes use of proteins which are embedded into the membrane. It makes use largely of these integral proteins. They have very complex structures. And this, at its simplest, what we're going to have a look at, first of all, are systems just of diffusion. Now, diffusive processes always operate from an area of high concentration to an area of low concentration. But as we've seen here, diffusive processes actually across the membrane are very inefficient. And very often the cell actually needs a constant supply of whatever it is, ions or whatever. And so the cell has channels whereby these ions and small molecules, etc., can move straight across the membrane and into the cytoplasm, always with the diffusion gradient from areas of high concentration to areas of low concentration, not the other way around. So here is, for example, this is a protein. It's a protein transporter. In this case, it's a non-specific transporter. It's going to transport, for example, it may transport a wide variety of anions across the membrane, something like that. It transports classes of substance across. And to do so, what is actually happening is that it's passing through a channel in the middle of the protein. That channel is actually very hospitable to the ion. It actually allows the ion to move across very, very easily. But other proteins are actually designed in such a way that they capture specific substances. So for example, glucose there is a specific glucose transporter. It's still transporting glucose from areas of high concentration to areas of low concentration inside the cell, but it is specific to the substance. And there are many of these specific for different substances. They usually bind the substance and they change in shape, and you can think of it almost as if the protein, the transporter protein, is swallowing the molecule and is spitting it out on the other side. But there's an important thing to note, that the energy for this is provided by this diffusion gradient. It's provided by the diffusive pressure, that this is concentrated on this side and this is of low concentration on that side. No other energy is expended. The cell does not have to. Once it's created this transporter, the transporter just works. It doesn't need any more input of energy as long as there's abundance of the substance this side and poverty of the substance this side. So that is the first thing. The other thing is that there is, I have told you this already, but it bears repeating, that the transport of water is done by special facilitated proteins. And these are called the aquaporins. Here they are here. They allow relatively free passage of water across. The only control which a cell can exert through the aquaporins is to actually get rid of aquaporins or rebuild them. Once an aquaporin is built, it just functions as a channel for the water. But the cell can exert some control by sometimes breaking down the aquaporin and taking it away. Or if it needs to transport water, building an aquaporins back. Not in prokaryotes. I'll give you an example, but it's not of a prokaryote. In kidney cells are very good at controlling the concentration of urine, for example. And one of the ways in which they do it is by continually controlling the number of aquaporins that they have in the cell membrane. So the aquaporins are water channels. Think of it as that way. All of these are diffusive processes, and they are passive. They are passive transport processes for the reason that we do not have to put in any extra energy. It's all provided by the concentration gradient. What we talked about, they're actually making it easier. So we refer to them as being facilitative diffusive processes. These proteins that we talked about, whether they're just channels like this, relatively non-specific, or whether they are the kind of that actually pump the molecule across. This is facilitated diffusion because it takes place more efficiently than natural diffusion. But there are many circumstances where the cytoplasm is going to require the transport of molecules against a concentration gradient. That is that if this is our cytoplasm here, the cell is going to need to move relatively rare molecules from an area of low concentration across to an area of already high concentration to accumulate them. This is very, very common. And you must, of course, realize that the cytoplasm is full. It's a solution of many, many, many different substances, biomolecules, ions, all sorts of things. And many of the things which are essential to it, it has to accumulate against a concentration gradient. Here, this is a transporter, a specific transporter, which actually works in quite a strange way. This transport may have to take place outside, towards the outside as well. The cell may need, for example, to pump substances out. This is a system called a sodium-potassium pump. You see these circles here? That is potassium. These here, this is sodium. And what this pumping system does is it opens this way here, and it pumps sodium in. And then it binds potassium here, and it pumps potassium out. So it's doing this. It's flapping back and forth like this, pumping one this way and one this way, one this way, one this. Every time it does, 2 inside, 3 out, 2 inside, 3 out. But this system is pumping ions against a concentration gradient. It is taking them to an area which is already high in concentration. And in order to do so, the cell has to put in energy. And it does so using the cell's primary source of cellular energy, which is ATP, adenosine triphosphate. We'll learn all about adenosine triphosphate when we talk about cell metabolism. So this system here requires a continual input of energy in order to carry on pumping things against a concentration gradient. All right, so I tell you what, let's take a 5-minute break. It's 4 to 5 now. So just take a 5-minute break, and then we'll come back and carry on. Don't get onto your cell phones. Rather get up, stretch, do something, and do something to get your brain moving again. What did I just do? I did something silly. How do I get back? How do I get back to where I was?
I can't hear you. I don't know if it's just my audio, but I can't hear anything.
Yeah, Professor, we can't hear you.
Yeah, I think it's muted.
Professor, we can't hear you.
I'm so confused.
Professor, he's still going.
Do we all just leave and come back?
There we go.
Can you hear me now?
Yeah.
Yes.
Oh, all right. I'm sorry. I don't know what was going on.
Yeah, it's okay.
Can everybody see the screen? Okay, cytoplasmic element.
Yeah, it was all good. It was just your audio was kind of like.
Okay. All right. Thank you. Sorry to hold things up. So what we're going to do now is we're just going to spend a little bit of time talking about the constitution of the cytoplasm itself, remembering that in the prokaryotes, there is no other membrane apart from the plasma membrane that surrounds it. So all of the things that we're going to talk about, it's a temptation to say they lie loose inside the cytoplasm. That gives the wrong idea, though, because in fact, the cytoplasm is extremely carefully organized. It's not that things are just random inside the cytoplasm. They aren't. And the first thing is a case in point. And that is that one of the most prominent features, of course, inside the cytoplasm is the bacterial chromosome. The bacterial chromosome is a circular thread of DNA, extremely long, in actual fact, compared to the size of the bacterial cell. If we took an E. coli cell, Escherichia coli cell, and you took the whole bacterial chromosome out and you spread it out, it would be about a millimeter or more in diameter. That's how much, and that's an enormous amount of DNA. So we've got to fit that into a very, very small package, right? The small package is the size of the bacterial cell. So that chromosome is very, very carefully wound and packaged inside the cytoplasm in an area that we call the nucleoid. And the nucleoid may, in fact, be so dense that it is actually visible. It's very easily visible. I showed you once before on electron microscopy, but sometimes you can actually see it even with with a good light microscope. You can make out the nucleoid in the cytoplasm. The second thing we've mentioned before, included in the cytoplasm, are small circles of DNA called the plasmids. And these are outside the chromosome, so they're extra chromosomal. And the plasmids often carry extremely useful information. They may carry antibiotic resistance. They may, often we find that the plasmids confer pathogenic capability to otherwise non-pathogenic bacteria. And the important thing about the plasmids is they are infectious. They can be moved from one bacterium to the other. This occurs naturally amongst populations of bacteria, but it's also a feature which we make use of, because when we, in genetic engineering, we make extensive use of the ability of bacteria to take up plasmids. Okay, the second thing, prominent in the cytoplasm, if you are able to visualize it, for example, with electron microscopy, not if you look at it under the light microscope, they're too small, but scattered throughout the cytoplasm are many, many ribosomes. And the ribosomes are sites of protein synthesis that we're going to hear a lot about later on. When we talk about bacterial genetics, we'll hear a lot about the function of the ribosomes. And the ribosomes, in actual fact, are made up of a mixture of protein and of RNA, ribosomal nucleic acid. And there are several kinds of ribosomal RNA that go to make up the ribosome. And one of the really interesting features of ribosomal RNA is that it has enzymatic ability. So when you're probably used to hearing that enzymes in cells are proteins. Well, most enzymes in proteins, most enzymes in the cell are proteins. That is true, but there are some enzymes which are not. And this is an example. Ribosomal RNA has enzymatic ability. We refer to that as being a ribozyme. So it's ribozymal activity. Don't worry over much about this sizing. This S refers to how fast they sediment when you centrifuge them. This is just to make the point that in the cytoplasm, the ribosomes actually exist in two halves. There is a large particle, large subunit, and a small subunit. Here's the large one here, and here's the small one here. And they only get together when we present them with messenger RNA. As soon as the ribosomal particles see messenger RNA, first of all, the small ribosomal unit binds to it, does certain things, and then the large subunit comes and binds onto and forms the complete ribosomal particle, which then performs the action of translation, of producing protein from the messenger RNA. Apart from that, inside the cytoplasm of prokaryotes, there are many, many, many other kinds of inclusions, that is, large collections of molecules doing certain specific things. You don't need to remember all of them. I've underlined 4 that I particularly want you to remember. But first of all, this is very common. These are called metachromatic granules. These are phosphate compounds, which are metachromatic because they appear in different colors under the light microscope. And these are stores of phosphate. Phosphate is very important to bacterial cells, or all cells, but especially to bacterial cells. and it is sometimes rare in the environment. So it is of great advantage to prokaryotes to store phosphate. They also are able to store polysaccharide, and just like us, for example, they are able to store glycogen. And this also will appear as granules within the cytoplasm. There are other polysaccharides that they can store as well. You may sometimes see large collections of oil, of lipid, mostly there for energy. And in certain bacteria, it depends on the type of metabolism. We'll talk about it later on. There are certain bacteria which actually, instead of using water, H2O, in their metabolic processes, they can use H2S, hydrogen sulfide. And they use the hydrogens off that hydrogen sulfide, and they accumulate sulfur inside the cytoplasm, more or less as a waste product. So I would ask those of particular importance. In specialized bacteria, for instance, in the cyanobacteria, which are able to perform photosynthesis. Photosynthesis is a very complicated metabolic process. And there's one part of it where the cell will take carbon dioxide and will actually bind it to water. It uses a huge amount of energy, but it has to be done under very specialized conditions. And that in prokaryotes is done in a special unit called the carboxysome. And carboxysomes actually are sometimes quite easy to see because they're very big, sort of hexagonal inclusions inside photosynthetic bacteria. Apart from that, just for interest sake, some prokaryotes, aquatic ones especially, have gas vacuoles. Now, these are interesting because they actually have a kind of membrane around them, but the membrane is protein. It's not plasma membrane. It's protein covering around the gas vacuole. And these are mostly aquatic bacteria, and they are to regulate buoyancy. The last one here is really interesting. This is called a magnetosome. And here it is here. All down here, it's a little bar of iron. And it is actually magnetic. That's why it's called a magnetosome. It's actually iron oxide. It's not as magnetic as raw iron, but it is still magnetic. And this has been a great mystery to people as to why these occur. And there are actually two functions which have been elucidated for this. The first is that it actually does act as a little compass, and it allows the cell to orient itself, especially prokaryotes, bacteria which inhabit the substrate. If the sediment, for example, is stirred up and they get swept up into the water column, they have to find their way back down to the sediment. They use this little compass in order to find their way down to the sediments again. But it appears that they have some respiratory function as well, and it is protective. During respiration, the process of respiration, cellular respiration, this substance, peroxide, is produced, sometimes produced. And peroxide is very dangerous. And cells have a number of different ways of dealing with peroxide. And it's believed that this actually functions to break down peroxide as well. All right, here's a really important one. In all sorts of ways. You will definitely see this in lab. And it is a feature which is peculiar to two particular genera of bacterial rods, gram-positive rods. And these two genera are Bacillus and Clostridium. both of them extremely important in terms of clinical microbiology. Bacillus also very important environmentally. And this is the production of endospores. Now, endospore means spore inside. This is to distinguish it from things, for example, like the fungi, which produce spores which are dispersed to the outside, and they're built on the outside. Endospores are actually specialized bacterial cells, and they are not reproductive cells. Normally, a bacterial cell grows, and when it reaches a certain size, it divides equally and produces 2 daughter cells, which then increase in size. But especially when conditions begin to deteriorate, especially when nutrients start to become impoverished in the environment, and when waste products begin to increase in concentration, when conditions begin to get harsh and growth starts to get difficult, reproduction starts to get difficult, these cells, what they do is they divide unequally. So here's the mother cell there, and here's the daughter cell there, a small daughter cell here at the end. But this daughter cell is treated differently. It doesn't just develop the normal cell wall. Instead, a highly specialized cell wall called the spore wall is developed around the spore, and the spore has very special properties. The spore, first of all, is almost completely inert. It has almost no metabolism at all. All of its proteins, nucleic acid and everything, are protected. They are carefully wrapped around specialized proteins. This wall, the spore wall around the outside, has special compounds which protect the inside of the cell from heat, from adverse conditions. And in clinical terms, this is very, very important because the spore wall also protects the inside, the cell, from things like very powerful disinfectants. These spores are also completely resistant to boiling. So this means that if you're dealing with Bacillus or Clostridium, which has produced these spores, and you are trying to sterilize something, it is no good to just boil it because the spores will persist. The vegetative cells, the cells undergoing normal activity, they will die off. They can be killed off easily with disinfectants or boiling, whatever, but the spores persist. And eventually these cells, the mother cell will die and release these spores. They're extremely small. They're very easily dispersed widely in the air, for example. And this is one of the reasons why both of these, Clostridium and Bacillus, are very important as contaminants. They can very easily contaminate wounds. They also very easily contaminate equipment, hospital beds, hospital surfaces, etc., etc., etc. And they are very, very difficult. The spores are very difficult to get rid of. Once the spores arrive in a suitable environment, then they germinate and they form vegetative. They grow very rapidly. And then if conditions become difficult again, they again produce another round of spores. Another feature of these spores is that they're extremely long-lived. And in fact, there are claims that spores have been revived, for example, from Egyptian mummies, thousands and thousands of years old. The reason for that is there's almost no metabolism. They are almost completely inert until they're placed into suitable conditions again. These spores are a source of tremendous problems in clinical settings. If you want to get rid of spores in. If you want to truly sterilize things, you want to sterilize equipment, you want to sterilize media or whatever, the media have to be heated up to about 120 degrees Celsius and kept there for about between 12 and 20 minutes. And in order to do that, to raise the temperature of a liquid above 100 degrees Celsius, you have to put it under pressure. So we effectively use a pressure cooker, which we call an autoclave. And it is routine clinical practice to autoclave media, to autoclave equipment, etc. Surgical equipment, all of those things are all autoclaved in order to sterilize them, to kill the spores that might be on them. All right, so let's have a look now. We've done the inside. Let's have a look now at some of the other features of the outside. And these are features actually out exterior, even to the cell wall. The cell wall is not necessarily, or the outer membrane in the gram negatives is not necessarily the only thing that lies outside the cell. There are other things as well. The first thing is called the Glycocalyx. And the Glycocalyx it depends on the bacteria and it depends on their circumstances, their growth circumstances. It can have different compositions. Sometimes it can be, in certain bacteria, it can be visualized as a very distinct thick layer around the outside. You will see this in lab. These dark dots here, these are actually bacterial cells stained probably with a gram stain. So they're a deep dark color. Then this outside here, the background has been stained. But surrounding the cells here, you see this clear area. This marks the area where there is this kind of slimy layer of polypeptide and polysaccharide. You can think of it as being like mucus. In the case of the capsules, it's relatively thick and well organized. And but sometimes it's very loose and kind of diffuse. I'd put it that way when you visualize it. And then it's referred to as being a slime layer, but they're basically the same composition. It's just that they're differently organized. This capsule can be a significant feature of pathogenesis because the capsule, the Glycocalyx, performs several functions. First of all, its primary function is that of attachment. Bacteria grow especially well once they're attached. And in pathogenesis, very often, the first stage of infection is attachment of cells, attachment of bacterial cells to a host cell. In environmental systems, attachment of bacteria to a surface is often required before they will start growing properly. That attachment is done in part by this capsule. The second feature of the capsule is, especially in pathogenic bacteria, it provides protection from the immune system. First of all, it prevents direct access to the bacterial cells. Second of all, it often protects from ingestion by things like the white blood cells, for example, by the leukocytes. And it may protect from antibody and all sorts of other things. So it provides immune protection. That's the first thing. The second thing is, the third thing is that you see how these are beginning to get together. The capsules are starting to attach to one another. The capsules and slime layers allow bacteria to build on one another and to form a biofilm. Very often of many different kinds of microorganisms, all wound together by this biofilm, which makes it extremely difficult, for example, in clinical settings, it makes it very, very difficult to access the bacteria that are on the inside. So these biofilms are very important environmentally. That's one thing. But second of all, in clinical settings, they are also very important. Not only on our own tubing. You can picture the urethra being infected, for example, with bacteria causing a urethritis. But you can also picture the bacteria forming biofilms inside catheters, for example, or things like that. So these, the formation of biofilms and the function of these capsules and slime layers can be extremely important. Okay, so I think I'm just going to stop there for today. Are there any questions before I finish? Okay.
Can I talk to you after class? I
Sure.
just started this class and I didn't like know of any like policies or procedures before, so.
Okay, everything is very clearly outlined in the syllabus. So have you read your syllabus yet?
I did read. I just had a couple questions about
That's
the
fine.
syllabus.
That's fine. That's fine. Yep, for sure. If you can ask the questions now if you want, or if you just want to meet in private.
Well, I don't know. I mean, I can just ask them after everyone else leaves.
Okay, fine. Anybody else? No, I don't know how to do this. Nielsen, how do I set up a private meeting with you?
Oh, we can just go after it.
Huh?
He can stay and we can just leave after.
Oh, seems a bit rude. All right then. Okay, everybody except Nelson. I'll see you then later on. Okay.
Okay.
I'll
Thank
see you next time. Yeah. Bye bye. Nelson, I'm just going to stop the recording. Okay. I'm getting